Morning, everybody. This is the walkthrough video for tax day. Um, Hello, cheers. I hope we'll have yellow bell work by the time that this runs. Hey, how are inducible and repressible? It's not Monday. For the love of Pete, it is Tuesday. Hi. Okay, so how are inducible and repressible operons the same, and how are they different? And I strongly suggest that you make a little table that looks like this. So will you pause the video if you have been in class the last couple of days, or if you have completed the assignment you for yesterday, you should know the answer to this. All right, so here are some of the ways that they are similar. There are lots of genes on one stretch of DNA. They have a protein that controls whether or not the RNA polymerase can bind to start transcription. And there's binding of that protein being controlled in these cases by a specific molecule, that's related to that process, right? So here are some ways that they are different, right? So an inducible operon is a transcription turned on by a molecule. That's the inducer. In contrast, a repressible operon is one that's turned off by a molecule, by a co-repressor. And both of these, as you'll remember, are different from a signaling molecule comes in from the outside world, like a hormone, for example, to control changing gene transcription translation. It is still Tuesday. Good. All right, cool. So today we're going to think about interactions that regulate eukaryotic gene expression and why that matters, because it's vital to so many processes. So a little heading for you, we're going to think about mechanisms that allow cell specialization. Last class, we talked about response to changing environments when we were thinking together about inducible and repressible operons, but we're really going to think about multicellularity together here. And again, this big question is not how it happens. We learned that 6A, but when it happens. And again, our focus today is on the complexity of anatomy and how it gets made. So Justin Timberlake, but bio. Enter the stickleback. So you'll remember from the work that you did while I was absent recently that stickleback are a really interesting organism to study the formation of spines. And you did a Mendelian genetics task about them. So what we're going to talk about today is baby's first cell to blastula, to one of these is the gastrula. I don't know enough about embryology to be able to make full fish, right? And that process, I mean, like you, fish are complex. They have a lot of different parts, a lot of different organs. They work in concert. They are exquisitely patterned. And so if you didn't watch it last time, you probably should watch the fish movie. But I wanted to remind you that the story we're going to be telling today is about the genes, the mutations in stickleback that cause the spines to change are not actually in any structural protein, but in a regulation part of the gene. So a reminder, transcription factors are these pinkish proteins here. So it says one, activator proteins bind to pieces of DNA called enhancers. They're kind of upstream, like the gene goes this way in this particular case, right? So their binding causes the DNA to bend. This brings them near a gene promoter, even though they may be thousands of base pairs away. And then the enhancers are the DNA sequence, and the activator proteins bind there, and they recruit these other transcription factor proteins to join them. That's the protein complex, which binds to the gene promoter, and in doing so, changes the binding environment for RNA polymerase to bind and move forward. There's some other things called insulators. I don't know if we've talked about the methylation as a control of DNA, but you learned about that in the videos. It's like a really important thing. So the proteins in the middle, we sometimes call mediator proteins. So you should be looking at page 150 in your review book, or if you don't have your review book and you need to use a textbook, in page 311 and 312. And you will need those for this particular feature. So, hey, after you've done today's assignment, you will be able to answer this question, and it'll help you see lots of different language on the AP exam. And this is to help prepare you for that. So hope you're doing well. Happy Tuesday, fake Monday. We'll see you soon. Bye.